Okay, welcome to week 5. So, in this week, we are going to talk about proteins. So, in last week, we actually talked about the blast, we talked about pairwise and the multiple alignments. So, basically, the computer lab number 1 last week was on blast. So, this week, computer lab number 2 will be focused on pairwise alignment, which is the alignments between the two sequences. It can be the gene sequence or it can be the protein sequence. And we are also going to do the multiple alignments. But the lecture today is on proteins. So next week, so this week and next week is basically proteomics week. So in the lecture, we are going to talk about proteins, but the lab is on pairwise and the multiple alignment. So next week in the lecture itself, we will, so this week is basic knowledge about the protein, their structure. Next week, I'm going to talk to you about the various tools which we can do in the proteomics field, where we can make the models of proteins. We can find different domains and motifs in the proteins. We can find the location of the protein. We can find if the protein has a transmembrane region or not. So all these tools I'm going to teach you in the lecture, and then we will have the computer lab number 3 in which we will apply all those tools and we will do various exercises. And before I start talking to you about proteins, I would like to let you guys know that I have posted the instructions for the final project, that what you're supposed to do with your final project. And I have also assigned the projects to every student. There are 2 documents on Blackboard in week 5 folder. There are instructions how to prepare for your final project, and also there is one document which says the list of the final projects with the name of students or something. So in that folder you can see which gene has been or which protein has been given to which student. So your name is there and the name of the project is there, the name of the gene or protein is there. So basically this week we will do like we are doing computer lab number 2. So we and the next lab is computer lab number 3. So by that time we are almost done with this course. So 7th week is basically we are going to discuss the research paper, how all these tools of bioinformatics they can be used in research, in the research work. We are going to discuss that, and you will be at home, you will be doing one experiment which is in the Carolina kit. So basically after the computer lab number 3, you have learned almost all the computer tools, and I have given you the name of the gene. You can start working on your project, and I would like all of you to, first of all, whatever gene has been given to you, first of all go on NCBI and find the accession number for that gene, because if you choose the wrong one, then your whole project is going to be wrong. First of all, please get your accession number checked by me. Once I tell you your accession number is correct, you can start working on your project. So send me by email the accession number for the gene which has been given to you. Go on NCBI site, look for the accession number of the gene which has been given to you. You have to use the database nucleotide, and you have to look for mRNA sequence, and you have to look for complete CDS. These are the 3 most important things. Database should be nucleotide, it should be mRNA sequence, because we don't want codons, so we don't look for DNA, we look for mRNA. It is basically the cDNA sequence, and you have to look for complete CDS. That is what you need to do. So this will be the very first step towards the project, and very important step, because if you go wrong here, your whole project will become 0. And this project actually will make you it will give you confidence that you know bioinformatics, because With this project, you will be applying whatever you have learned till now. You will be applying that knowledge to work on your project, to find out about this gene or protein I have given to you. And that is basically the real application of the knowledge of bioinformatics. Whatever you have learned, when you will try to make your project, you will yourself feel that you are actually learning a lot while doing this project. So it's very important. So my best wishes to all of you for that. And anytime during when you're working for this project, anytime you face any difficulty, and you will face like when you work independently, you will find you will have some problems, you're most welcome to contact me. Don't go wrong, but consult with me. I will be there to help you, even if you wake me up at midnight. Okay, so make sure you do this project very nicely, and it should be the best. Okay, so let's start today's lecture. We're going to talk on proteins today. So we have talked about the process, the gene expression. In the very first lecture, we talked about how gene codes for a protein. We have talked about transcription, translation. You know that the sequence in the mRNA, there are codons, 3 ribonucleotides, they code for 1 amino acid. And these amino acids, they are brought by the tRNA, and they are brought by the transfer RNA, tRNA, and tRNA has an anticodon which recognizes the codon on the mRNA and brings a specific amino acid. And these amino acids, they together form, because there's a formation of peptide bond between them, they form the peptide chains, and they formed on ribosomes, and that process is called translation. Today we are going to learn about we'll talk about the structure of the protein. Okay, so proteins, as we know, the DNA codes for proteins, and these proteins are very, very important, and the majority of the functions in the cell are performed by proteins. Proteins in our cells, they're doing a lot of functions. So first of all, let's have a review on the different functions which are played by the proteins. Number 1, enzymes. Majority of the enzymes we know they are protein in nature. So enzymes, I assume you know what enzymes are. Enzymes are the ones which make the rate of reaction fast, which speed up the reactions. They act as catalysts. So we have a lot of enzymes in our system, and they are mostly proteins which are making all the reactions going on in our body fast. The proteins, they can be storage proteins. As you know, in the egg, people eat egg because they want to have they want to make their muscles, they want protein, because egg has a lot of storage protein, and it is called albumin. So there is a protein called albumin in the eggs, which is the kind of a storage protein. Another example of storage protein is milk. Milk has a protein called casein. Then the proteins are defensive proteins, which means basically the proteins, the antibodies. If you have heard about antibodies, like when we get infected by some bugs, then our cells, they produce our special cells, the cells which belong to our immune system, they make special proteins called antibodies, and they help us in they give us protection from all the bugs which invade our cells. So they act as defensive proteins, they protect us from diseases. Transport proteins, all our cells, they're surrounded by a membrane. So in the membrane, there are some proteins embedded which help in the transport of molecules or solutes across the membrane. Like things can move into the cell, or things can move out of the cell, and all these things happen through the transport proteins. So here is another role of proteins. Another very important transport protein is hemoglobin. You must have heard about hemoglobin. Hemoglobin is a protein, and it is a carrier of hemoglobin, is present in our red blood cells, and it can carry oxygen throughout our body. Okay, hormonal proteins. So the hormones, lot of hormones, They're protein in nature. Like insulin is a hormone. It's a protein in nature which is responsible for the regulation of blood sugar level. Then contractile and motor proteins. As you know that the muscles help muscles contract and relax, and because of that we can do various movements. And these muscles, they are made of proteins called actin and myosin. Then receptor proteins. So there are a lot of proteins which are present outside our cells. They are embedded in the membrane. They basically receive signals from the environment, and when they receive signals from the environment, then they can cause various changes inside our cells. And there are some receptors which are built into the membrane of nerve cells, and they detect signaling molecules released by the other nerve cells. So as you know, our nerve cells, they help in the conduction of the impulses, and that is all because of some of the receptors which are present on the surface of some of the nerve cells, and they receive signals from the neighboring nerve cells. Now structure proteins, there are a lot of proteins like keratin, which is in our hair, which is in our nails. These are all example of some structure proteins. Collagen, elastin, these are all proteins, and they provide a fibrous framework in animal connective tissue. So these are all examples of structure proteins. So the proteins in our cells, they do a lot of functions. So the majority of the functions in our cell, they are performed by proteins, and DNA codes for proteins, and you know the process. Now why, so in this lecture we're not going to talk about transcription and translation. We're going to talk about the structure of protein. Now the structure of protein is at 4 different levels, and that's what I'm going to discuss with you. So why it's important to know the structure of protein? It's very important to know the structure of protein because there is a structure function relationship. So the function of any protein depends on its structure. So proteins are fundamental components of all living cells, performing a variety of biological tasks which we just discussed. Each protein has a particular 3-dimensional structure that determines its function. So the function of any protein is dependent on its structure, and the structure of protein is at 4 different levels. So every protein has a 3-dimensional structure. That is what we are going to discuss today, and in the 6th week we are going to make the models of proteins by using bioinformatic tools. Okay, protein structure is more conserved than protein sequence and more closely related to the function. So there is a big relationship between structure and function, and to know the function of protein, it's a very important thing to know what the function will be done by the protein. And these days we can we can have a guess on the structure of protein by knowing its structure with the help of the bioinformatics. So let me talk to you about the structure of protein. So basically, as you know, the proteins are made of amino acids. Proteins, like we have already discussed in DNA, also DNA is a polymer, which means it's a long chain, and it is a polymer and it is made of monomers called nucleotides. So similarly, proteins are also polymers, and they are made of monomers called amino acid. Okay, amino acid. Now let's see the structure of this amino acid. So this amino acid, it has one carbon here. It is called a alpha carbon. Okay, this carbon, the carbon has valency 4, which means it can bond with 4 different atoms or 4 different groups. So this is the basic structure of an amino acid. Here it has the carbon and its valency is 4, so which means it can bond with 4 different groups. So you can see here is a group called COOH group, and the second group is NH2 group. This is called amino group. And also third group is R group, or it's called the side chain, and fourth is the hydrogen atom. So this is the structure of basic amino acid. There are 20 different amino acids. Okay, there are 20 different amino acids, and all amino acids, they have a basic structure. And what is the basic structure of the 20 amino acids? This is the basic structure of any amino acid, which means that all the 20 amino acids, they will have a carbon, and that carbon is called alpha carbon, and this will be attached with 4 different atoms or groups. So it is attached with hydrogen, it is attached with carboxyl group, it's attached with amino group, and it is attached, the fourth one is R group. Now in all the 20 amino acids, these 3 groups will be same, but what makes one amino acid different from another? It is this R group. So in 20 amino acids, they all have carboxyl group, they all have amino group, they all have hydrogen, but they have this R group which is different. And I can show you. So this is the general structure of amino acid which I just said. This is again same thing. These are 20 different amino acids. Okay, you can see here all the amino acids, they have, this is the carboxyl group, this is amino group, this is hydrogen, carboxyl group, amino group, hydrogen, carboxyl group, amino group, and hydrogen. You can see here all the amino acids, they have these 3 groups same, but what makes them different from each other is this R group. So you can see here, this is the amino acid called glycine. It has, the R group is H, only H. This is glycine. This is alanine. The R group is CH3. Valine, it has this R group. Leucine, it has this R group. Isoleucine, it has this R group. So you can see here, there are 20 different amino acids. They all have same basic structure, but what makes them different from each other is the presence of a different R group. Some of the amino acids, they are negatively charged. Some of them are positively charged. These ones are the ones which have polar side chains. These ones are the ones which have non-polar side chains. So these are the negatively charged, and they have the COH group, and these are the positively charged, they have NH2 group. Okay. Now, as I just said, the proteins, they are polymers, which means they are made of monomers. And what is the monomer of a protein? It's amino acid. So if we look about DNA, DNA was also a polymer. It was made of monomers. The monomer of DNA was nucleotide. And remember that those nucleotides were linked with each other. It's very important in a polymer, it's very important in a polymer for all these monomers to link with each other, otherwise they cannot form a polymer. So in proteins, these amino acids, they are linked with each other. They are linked with each other and they form a bond which is called a peptide bond. Now how do they link with each other? Let's see here. Now this is your one amino acid, this is your second amino acid. Now how they're going to link with each other? This is the carbon, this is the carboxyl group, this is the amino group, this is the R group, and this is the R group. So, this is carboxyl, amino, this is hydrogen, this is the R group. This is our second amino acid, carboxyl, amino, hydrogen, and this is the different R group because it's a different amino acid. But now the question is how these amino acids, they're going to link with each other. So to gather these 2 amino acids, they're going to lose 1 molecule of water, which is called a dehydration reaction. So this amino acid is going to lose OH, this amino acid is going to lose H, and together they lose 1 molecule of water. And together when they lose 1 molecule of water, that results in the formation of a bond between these 2 amino acids, and this bond is called the peptide bond. And this is the way the different amino acids, they link with each other. Suppose these 2 amino acids, they have formed a bond with each other. Now here we have another amino acid. The same thing is going to happen. Okay, this is the way these amino acids, they link with each other. They together, 2 amino acids together lose 1 molecule of water. That results in a bond. And the structure of protein can be studied at 4 different levels. It is called primary structure, secondary structure, and tertiary structure. And the 4th level is called the quaternary structure. Now the important thing to know is what are these structures. Now first of all, what is the primary structure? Now primary structure is basically the sequence of amino acids. So how these amino acids, they are linked with each other, which amino acids. So as you know by now, there are 20 different amino acids, and these amino acids, they can link with each other. Okay, so now what is the order of the amino acids? Which amino acid is number 1, which is number 2, which is number 3? The order of amino acids, like the first one is proline, second is valine, third is cysteine, fourth is leucine, fifth is isoleucine. Or there can be another protein in which number 1 is alanine, and the second one is proline, third one is valine. So what is the order of these amino acids, because there are 20 different amino acids, gives the primary structure to the protein. So primary structure is the order of amino acids. Now if you see here how these amino acids, they're linked with each other. Okay, so this is, here is a peptide bond. Okay, here is one peptide bond. This is first amino acid, this is second amino acid. They're linked by peptide bond. This is second amino acid, this is third amino acid. They're linked by another peptide bond. Okay, so this is the peptide bond. At the peptide bond, you can see here there's one oxygen and there's a hydrogen. At second peptide bond, again you can see there is a oxygen, there is a hydrogen. Okay, so at every peptide bond, you can see there's a hydrogen, there's an oxygen. So important thing to know is oxygen is very electronegative in nature, which means that it can pull electrons towards itself, or in a simple terms, in a simple way, the easiest way to understand this concept is you can just think like oxygen has a little negative charge on it. Okay, oxygen has a slight negative charge on it, and hydrogen has a slight positive charge on it. And these oxygens and hydrogens, they are at every peptide bond. They are here, and if this is this amino acid and there's next amino acid, there's again a peptide bond, then there's this amino acid, there's a peptide bond. So these peptide bonds, if this is the primary structure of a protein, these peptide bonds are there throughout, because these peptide bonds are between every amino acid, and at every peptide bond there is negatively charged oxygen and there is positively charged hydrogen. So do you think if there are negatively charged and the positively charged atoms, do you think they can attract to each other? Yes, they can attract to each other. They can attract each other. They can be attracted towards each other, and because of their attraction, this polypeptide chain, which is made of various amino acids, it can fold, and this folding is because of the attraction between these oppositely charged amino acids. And when this chain folds, it gets this folded chain because of the attraction between oxygens and hydrogens which are present at the peptide bonds. This chain is going to fold, and this is called the secondary structure. And I would like to tell you here, when the chain folds, because of the oxygens and hydrogens which are at the peptide backbones, they can acquire two kind of structures. They can fold like this, and they can attain this kind of structure. This is called alpha helix, or they can also have these kind of structures. Okay, these are like parallel sheets, and these are called beta pleated sheets. So this is kind of when the chain folds like this and it forms like a coiled structure, this secondary structure is called alpha helix. And when the chain forms these kind of sheets which are like parallel to each other, this kind of secondary structure is called beta pleated sheets. So when the chain has folded like this, okay, it has either alpha helix or beta pleated sheet. Remember that if you see this peptide here, so this is first amino acid, this is second amino acid, this is third amino acid, right? And these are the peptide bonds. So they have oxygens and hydrogen, but do not forget that every amino acid has this. You can see here, this is amino acid number 1, this has this R group. This is amino acid number 2, this has this R group. This is amino acid number 3, this has this R group. So now in the polypeptide chain, if it has folded, it has either formed alpha helix or beta pleated sheet, but this chain is also rich in these R groups. So because these R groups, they can also interact with each other, and these R groups can interact in various ways. And sorry, I forgot to tell you one point here. The secondary structures, when they are formed like this and this, they're always because of hydrogen bonds, because these are the bonds which are formed between oxygen and hydrogen. Okay, so this folding of the chain alpha helix or beta sheets, which is secondary structure, is because of hydrogen bonds. But these kind of R groups, they can interact in various ways. They can form ionic bonds, they can form disulfide linkages, they can form hydrophobic interactions, van der Waals interactions. So basically what I'm trying to say, when these R groups, they are going to interact with each other, they can form various bondings, and because of which this chain, this folded chain, is going to fold more. This has already folded, and because of the interaction between R groups, this chain is going to fold more, and that kind of structure is going to called a tertiary structure. If the protein is made of only one polypeptide chain, then whatever structure this folded chain attains is the three-dimensional structure of a protein. A point to know here is there are various kinds of proteins. There are various kinds of proteins, and these proteins are made of different number of polypeptide chains. There are some proteins which are made of only one polypeptide chain. There are some proteins which are made of two polypeptide chains. There are some proteins which are made of three polypeptide chains. There are some proteins which are made of four polypeptide chains. Now, if a protein is made of only one polypeptide chain, then how does this polypeptide chain folds and attains a tertiary structure? That is the three-dimensional structure of a protein. But if a protein is made of more than one polypeptide chain, so then if suppose there's a protein which is made of two polypeptide chains, those two individual polypeptide chains will have a primary structure, which means whatever is the order of the amino acids. Every polypeptide chain is going to fold and have a secondary structure, and every polypeptide chain is going to further fold because of the interactions of R groups, and it is going to have a tertiary structure. Now, if a protein is made of two polypeptide chains or three polypeptide chains or four polypeptide chains, how does these fully folded polypeptide chains who have attained tertiary structure, how does these fully folded polypeptide chains which have attained a three-dimensional structure, how do these chains further interact with each other? That gives a structure that is called a quaternary structure, and that is the final three-dimensional structure of a protein if that protein is made of more than one polypeptide chain. Otherwise, if it is made of only one polypeptide chain, then this tertiary structure is the three-dimensional structure of a protein. But if it is made of more than one polypeptide chains, because these fully folded polypeptide chains are going to further interact with each other, and then they are going to acquire this fourth level of the structure, which is the quaternary structure. So let's see. So this is the primary structure, this is the secondary structure, this is the tertiary structure, and this is the quaternary structure. So quaternary structure will be possible only if a protein is made of more than one polypeptide chain. So here are some more examples. You can see this is your secondary structure. They can be alpha helix, beta pleated sheet, and these are folded chains. These folded chains can further fold, and then they're going to have their tertiary structure. And if the protein is made of more than one polypeptide chain, you can see this is one chain, this second. How these chains, these fully folded chains who have already acquired tertiary structure, how they further interact with each other gives a quaternary structure. So this is the structure of protein at four different levels. Secondary structure is always because of hydrogen bonds, and tertiary structure and quaternary structure both are because of various kinds of bondings, and they can be hydrogen bonds, ionic bonds, hydrophobic interaction, van der Waals interaction, disulfide linkages. Because here the involvement is of the R groups. So there can be hydrogen bonds, there can be hydrophobic interactions, van der Waals interactions, ionic bonds, disulfide linkages. These are all different kinds of bonds which can form because of the interaction of the R groups. So this is example of a protein, very important protein, hemoglobin, which is carrier of oxygen throughout our body, and this is made of 4 polypeptide chains. It is made of 2 alpha and 2 beta. Okay, so this is the quaternary structure of hemoglobin. There are 4 polypeptide chains, 2 alpha, 2 beta. This is the tertiary structure of 1 beta subunit, and this is showing you the alpha helix. This is the secondary structure of the hemoglobin. So you can see here how they have formed these coiled structures, and primary structure is the sequence of the amino acids in the polypeptide chain, and this is the primary structure for the beta subunit. So that's all about the structure of the protein. It's very important to know the structure of protein because in the next week's lecture we are going to talk about various tools. We are going to actually make use of bioinformatic tools, and we are going to make the model of the structure of the protein. So if you know the sequence of the amino acids in a protein, with the help of the bioinformatics tools, we will try to find out what 3-dimensional structure that protein is going to have, which basically means if we know the primary structure with the help of the bioinformatics, we will be able to make the 3-dimensional structure. I'm sorry, we will be able to make the 3-dimensional structure of the protein. Okay, it's very important to know the structure, because if we know the 3-dimensional structure of the protein, we can predict its function. So that is the importance of knowing the structure of the protein. So that's all is the lecture on proteomics today for week 5, and we will continue proteomics in week 6, and you will learn more about it. Okay, thank you very much.